Okay, we're recording.
Okay, so
Cell cycle regulation.
for those of you that were super confused, like us, when it was explained.
All right.
So we pretty much mapped out all of G1 regulation. Okay, so we're going to start with mitogen. A mitogen binds to its receptor.
And mitogen is basically anything that like proliferates cell growth.
So the main one that was in our notes, I think, was insulin like growth factor.
Mm-hmm.
Is that correct? Okay. And then that activates RAS. RAS activates MAP kinase, and then the MAP kinase, that whole deal.
Yeah.
Leads to the activation of regulatory proteins. This eventually is going to lead to the transcription of the MIC gene, which gives us the MIC protein. So MIC is going to do three basic things, but we're going to kind of go through that in stages. So the first thing that it does is it activates the transcription of cyclin D, SCF ubiquitin ligase, as well as E2F. So we're going to talk about this cyclin D first. So cyclin D, once it's translated into its protein, is actually going to form a complex with CDK and forms the G1 CDK complex. But how do we get this complex, you might ask? So cyclin is going to come all the way into our thought bubble here. So here we have CDK, ATP is hanging out in it, and then we have the CDK T domain, and this is the inactive form of CDK. So once this whole cascade occurs and cyclin is made, cyclin comes in, combines with CDK, and you get a partially active complex. And it's partially active because their interaction changes the conformation, and you get this T loop that is like the T domain of CDK, and that interaction makes it partially active. And then to get it into its fully active form, CDK activating kinase, or CAC, phosphorylates cyclin, and then you have your fully active CDK cyclin complex. But there are some other things that regulate this. So some of the main ones that we talked about in class are these CDK inhibitors. Some belong to the INC family, and some belong with ChIP-KIT, I think we're calling them. So INC only inhibits a couple different types of complexes.
Yeah, it's basically anything in the cyclin D family.
Yeah, cyclin D1, D2, D3. And then ChIP-KIT pretty much do it all. And those ones are P21, P27, and P57, and they're all encoded by different genes. So for example, the INC4 family can come in and they can inhibit this step, which prevents CDK from joining with cyclin, and it inhibits this process right here. And then you also have this interaction with CDC25 phosphatase and WEE1 kinase.
It is a nicokinase.
Yes, so to inactivate this form, WEE1 kinase adds an extra phosphate group to this complex, and having two phosphates makes this complex inactive. To get back to the active state, CDC25 phosphatase takes out that phosphate, gets you back to the active form. So basically, that's how we got this complex here. So where would we like to move next?
Well, okay, so most of the time when the cell's not ready to like enter the cell cycle yet, this complex is inactivated by P27, which, if we refer back, is one of the ChIP-KIT, as we're calling them. So until something comes and takes it off, it's just going to be, ignore the phosphate for now, it's just going to be sitting there inactive.
Big hug.
Big hug, yeah. So once Mike transcribes the SCF, or as he likes to call it, science fiction, ubiquitin ligase, which takes the P27 off of the CDK cyclin complex by phosphorylating it and then ubiquitinating it.
Anonymous kinase phosphorylates it first, though.
Yes.
Right? Anonymous kinase. Like,
yeah. We looked
there's
it up.
Google Docs with anonymous,
But
like monkeys and stuff.
yeah.
So anonymous kinase comes in, phosphorylates it, and then that allows the ubiquitin ligase to ubiquitinate it and target it for proteasome degradation,
Yeah.
which gives you more of this active CDK cyclin complex right here.
And then the purpose of this CDK cyclin complex, now that we have it in its fully active form, is to regulate the master regulator of the cell cycle. So it's kind of a double negative. It promotes cell cycle going forward by inhibiting RB.
Should we bring in E2F at this point?
Yep.
E2F in here.
So yeah. So RB inactivates E2F, which is another thing that Mike transcribes. Now E2F wants to send the cell cycle towards the S phase party. But RB is kind of a downer on that party. So until we get CDK4 cyclin complex phosphorylating RB and having it go party by its alien self, E2F will be in RB jail.
Yeah, so E2F is in its active form when it's free, but when RB is active, which means it's not phosphorylated, the master regulator in its active form inactivates E2F. So it's that phosphorylation that releases RB to get us back to our active E2F. And once we have active E2F, it leads to S phase gene transcription, and then we get cyclin E, which is related, it's a G1S cyclin, and cyclin A, which is an S cyclin. And then moving on from there, you get a CDK2 and cyclin A complex, which is called active S-CDK, which eventually leads to DNA synthesis and the S phase party. So there's a lot of other regulation going on at this point too. So there's positive feedback from S phase gene transcription to make more active E2F, and as well as this complex here with cyclin E, CDK2, and active S, CDK, they're all feeding back to phosphorylate, to assist in phosphorylating more RB to get more active E2F to push the whole process towards the S phase.
So, should we talk about synthesis or when things go wrong?
How about when things go wrong?
Okay,
And then
we'll have Nathan.
Actually, it makes more sense to
Okay.
switch.
You want to switch?
Yeah.
Oh yeah, I want to go up there and call shirt. Let's talk about shirt.
Okay, so simultaneously into the G1 phase, you've also got another few players involved that are getting your DNA strand primed for replication. So,
Some of the steps here.
sure. So the steps that we have listed here, we're gonna have G1, you're gonna have steps 1 through 3 here. So these are all happening simultaneously with the rest of this. And then when S phase occurs, you're going to have step 4 and 5, which I'll walk you through. And then even beyond that, you're going to have the final step 6. So basically, you've got your origin recognition complex that is constitutively sitting on your DNA strand, not really doing anything.
Its nickname is T-shirt.
Its nickname is One Arm T-shirt. So once we have the mitogen binding and all this other stuff starts to kick into action, CDC6 comes. We think maybe through MYC, but not 100% on that, but it comes and binds to the sleeve of the shirt, and then you start to have MCM or little macaroni come in and form little rings around the ends of your DNA strands. So that's called the pre-replicative complex, and it's going to sit there primed and ready until you have SCDK, which we showed getting activated at the end of our E2F positive feedback loop, until we have enough of that, which is going to go ahead and push the whole thing into S phase. So once you've got enough SCDK, it immediately just kicks into action and basically as we understand it, phosphorylates everything. So it's going to phosphorylate our friend ORC, the little shirt. It's going to go ahead and phosphorylate CDC6, and it's going to phosphorylate our MCMs. So once Mr. Shirt gets phosphorylated, and once MCM gets phosphorylated, they're going to open up. So helicase, we assume, has come in here and helped to open up the strands so that DNA Paul can come in and start doing its thing. We've officially entered S phase, and at the same time, CDC6 has split. Now it's hanging out and waiting for SCF ubiquitin ligase to come in. Because we're kind of on a balancing act, we're kind of on a tightrope at this point, because we're concerned about, we don't want too much DNA. We only want DNA replicated one time. So basically, we don't want any more pre-RC complexes to get formed. In order for that to happen, we need to have degradation, immediate degradation of CDC6 and MDM. So, or just CDC6, MDM is going to have another way of getting rid of it. So SCF ubiquitin ligase comes in, and ubiquitinates our phosphorylated CD6, which causes it to head over to the proteasome, get destroyed, and after MDM is phosphorylated, at some point, we don't really worry about the specifics of this, but it's going to exit the nucleus and not form any more circles and not recruit any more helicase. So after all that has occurred, and DNA Paul has been allowed to do its thing and replicate the strands, we're going to have no more CDC6, no more MDM, so all you're left with is Mr. ORC, once again, sitting upon his DNA strands, alone, phosphorylated at this point, but inactive.
Okie doke. Alright.
So that's what happens in a typical normal cell. But what happens when things go wrong? So,
Tell me.
okay, so when DNA is damaged, normally P53 is just kind of hanging out with MDM2, just sort of in the cell, not doing much of anything. But when DNA is damaged by like UV radiation, it sends out a signal for protein kinase activation and phosphorylation of P53. So P53 dissociates from MDM2 and is phosphorylated to its active form. So once it's phosphorylated, it does many things, but the thing we care about right now is that it transcribes P21, which is a cousin of P27, and does pretty much the same thing on bigger kinase or bigger complexes. So it's transcribed, and then it inactivates cyclin A and cyclin E, which, if you remember, have the positive feedback effect and push the cycle into S phase. So if it's in the P21 jail, it cannot continue with its DNA cell cycle thing.
The party.
S phase party.
Can't join the party.
Yeah.
Cool.
And that's G1.
And that's G1 and part of S.
And
That was
DNA replication.
easy, right?